Bismillahirrahmanirrahim. Assalamu alaikum. In this lecture, we will discuss the topic of transplantation of tissues and organs. Grafting refers to a surgical procedure to move tissue from one side to another on the body. All body cells have unique antigens on them, so foreign cells transplanted anywhere into the body of a recipient can produce immune reaction. Actually, this tissue transplantation is the same procedure as we have in the blood transfusion. As we have different types of the blood groups, like that we have certain immune reactions, immune systems in our body. Some are generalized immune system and some are very specific to each and in every individual. If in any case any organ is going to be transplanted into the person, for that there are chances for the rejection, because this foreign organ that is going to behave as an antigen for our body. So, if it is foreign antigen and it is not suitable for our body, our immune response is going to react with that. And as a result, the organ rejection can take place. Only difference is that in the blood we have certain blood groups, and before blood transfusion we have to determine exact blood grouping, and also before transfusion we have to do the cross matching to avoid any complication, to avoid the rejection of blood coming from the donor. Same for the organ transplantation. Before transplantation, we have to check either it will be suitable for the person, for the recipient, or there are chances for the rejection of that specific organ. This whole takes place under a very specialized immune system. Before discussing that system, I would like to explain the types of the grafting. It may be autograft, isograft, allograft or xenograft. Again, in this type of the grafting, we have on one side we have the donor and on the other side we have the recipient. So, coming from the donor side, either this specific organ given to the recipient, either it is suitable or not. It is one thing. And secondly, what is the relation between the donor and the recipient? If the graft taken from one part of the body of an individual and transplanted onto another side in same individual, that is in individual, donor or recipient, the same one, not the other thing. Donor is the same person as the recipient. That kind of grafting is called autograft. Then graft taken from one individual and placed to another individual between, for example, the identical twins. That will be isograft, as its name shows. Iso means same. So same person other, that is twin. If you are taking the graft from the twin brother or twin sister, like that, then it will be the isografting. Then allograft, graft taken from one individual placed on genetically non-identical member of the same species, kidney, heart, liver and other things, bone marrow, that is named as allograft. Finally, we have the xenograft, in which the graft taken from one individual placed to another individual belonging to another species. In allograft, we have the same species. The individual is different, but the species remains same. But in xenograft, the species even changed. That is from animal to the man. Now through this figure I want to show you what is the autograft, isograft, xenograft and allograft, as I have already discussed all these things. This is the picture or figure just for you to remember all these types more easily. Now comes the specific immune system through which the organ rejection, grafting that can be rejected by the body, tissue typing, that specific immune system, a specific complex system that is named as human leukocyte antigen complex of antigens or HLA complex. This is the specific system through which we are going to check the graft coming from either type of these and it is suitable for the recipient or not. This is the basis for tissue typing and further the graft rejection. The most important antigen for causing graft rejection are a complex called human leukocyte antigen HLA complex system. 6 are present on tissue cell membranes of each person. Development of immunity against any of these antigens can cause graft rejection. And they can occur on the white blood cells, so tissue typing for that is done on membranes of lymphocytes. How we can prevent these things? Obviously, anything which is coming from one person, either to inject it to same person or coming from his or her twin brother or sister or the other, it has many chances for its rejection. But if the individual is same, the rejection chances that will be very less, that will be minimized, or the rejections chances that will be highest in case of graft taken from the separate species, from animal to the man. So it depends, rejection depends what is the donor and who is the donor and who is the recipient. In same individual, the chances for rejection is minimized. And if we have xenograft, the last category, in which the graft is taken from animal to the human, that has the greatest chance for the rejection. One thing. And the second thing, without immunosuppression in the normal person, even with the best possible tissue typing, allografts seldom resist rejection for more than a few days or weeks. Because T cells are main cells of immune system for killing grafted cell, their immune suppression is much more important. So what I am trying to show you, that is, before you are going to transplant any organ to the recipient, we can give the suppressant, immunosuppressant, to minimize the chances for its rejection. For that purpose, the therapeutic agents may be glucocorticoids, various drugs, cyclosporin, or immunosuppressive antibody therapy. On one side, you are suppressing the immune system for the rejection of these foreign transplanted organs. At that time, you must keep this thing in your mind that after given the immunosuppressant, the whole immune system will be suppressed and there will be great chances for the infection on the other side. If we have no rejection of the organ, but due to the immunosuppressant we are going to give the recipient, because of that the chances for rejection that is minimized. But after given the immunosuppression, the whole immune system that will be disturbed and there will be more chances for the infection. So go according to the condition, and for that purpose, to avoid the infection, we use the other therapies also. So we can use for rejection, to avoid the rejection, we can use these different therapeutic agents as I have already named. So with this, the rejection chances they will be minimized. And with this we complete our topic regarding the blood grouping and RH system, RH incompatibility, and this the last, that is the tissue rejection. Inshallah, next we will start the hemostasis. Okay, thank you and Allah Hafiz.